This is the first time I've seen this thing up at the top of the screen. It's just saying that the website that hosts the textbook will be down for schedule of maintenance at 2 p.m. on Saturday. So if you tend to do work involving getting to the book on Saturday afternoons, be aware that it'll be down. That's Central Time Zone because they're in Texas. So that would be, what, 3 o'clock here?
But,
So,
question.
yeah.
When the semester is over, is it like, how do I get the book? Like, everyone just review the book. Is it possible to do?
Well, you can bookmark it now, so you know where to get to it. You can download versions using the Get This Book button right here. Or if you forgot to bookmark it, go to my faculty webpage and there's a link to it on there.
Okay.
Yeah. Or you can just go to OpenStax College and you'll find my book there.
All right.
So, yeah, it's freely available, easy to get to whenever you want to. So I didn't finish stuff last week on neuroanatomy because we spent the whole time dealing with the excitable membrane stuff. So actually we're going to finish that today because it really does fit in with what we're doing here. In the more traditional lecture lab format, we have one lab where we do the tissue stuff and then another lab where we look at the brain models, which coincides with the weeks when we're doing lecture on the tissue and the electrical activity stuff. But in this format class, it didn't quite work out that way. So the neuroanatomy lab quiz includes questions on the tissues, which we actually did 2 weeks ago, I guess now. I should have gotten onto the anatomy stuff last week, but didn't have time to. So we're going to do that today because it fits in with the chapter that we're looking at. Now, actually, the chapter that we're looking, or the material that we're looking at today, is this chapter that says anatomy of the nervous system, but also some of the functional aspect of what's going on for this week is covered in the somatic nervous system chapter. I'm not going to refer to that chapter in any of the stuff that I'm doing today, but there is a bit that goes with that. Next week, we're going to be looking at the chapter that's called the neurological exam, which is to look at a lot of this stuff from a clinical point of view. And actually, some of what I'm going to be doing today is going to have a clinical side to it also. But we'll get sort of more into that and look at neurological situations in more depth with that. The part 1, 2, 3, and 4 assignments that you have for this week, that's in this week's folder, are mostly out of this anatomy of the nervous system chapter, but there are some functional aspects of it that cross over into the somatic nervous system chapter. So there might be some questions that are more out of that chapter than this first one. So be aware of that. But there are not assignments that go directly with the neurological exam chapter, which we'll be doing next week. Now, I did this material already for my night class last week and went a little bit long. Part of the reason for that is that this is my specialty. This is what the nervous system is what I studied in graduate school. And some of what we're going to be covering today is some of the most interesting stuff that I find from this. That I find students really get a lot of interest out of too. So I might be kind of inclined to go off on tangents even more so than I normally do, but I think it would be useful. With my night class, I had several students that asked questions more to their interest that got us off what the main topics were. I don't mind that. I think if you're interested in something, you're going to learn more than if you're just sort of following through in the course of what I want to cover. So if you do have questions that seem kind of tangential things here, that's fine. Now I'd like to start with considering what's happening in the embryo, because I think that's a good way to understand the basic layout of the brain here. Now, when I was in college, I took courses where I learned about the nervous system like this. I learned about basic anatomy like you guys are right now. But really my best or strongest memories of learning this stuff come from graduate school, because I spent more time doing it there. And in graduate school, I was actually working with the embryological brain a bit. So this, I think, is a great way to understand how the brain's put together. So I want to start here, and it's not unique to me. There are other sources that'll take the same approach. Now, the nervous system starts very simply as just a tube, and by looking at how it develops from that structure, we can get a lot of understanding of how things are connected and laid out in the brain. To start with it, though, I want to explain where the tube comes from. The nervous system, the embryo is made up of three major tissue types. There's endoderm, which is the inner surface of the embryo, mesoderm, which is the middle portion, and the ectoderm, which is the outer covering of the embryo. So what we're looking at here is just a section taken out of a kind of hollow, spherical, ovoid-looking thing here. That's the early embryo. Along the back of the embryo, the ectoderm, what's going to essentially give rise to the skin, folds in a little bit, creating this ridge going along the back. The two edges of the neural fold come together and seal off a tube, which we see labeled here as a neural tube. And so running along the back of the embryo, there's this tube. And that's the nervous system at its simplest. It has an anterior end, which is where the head's going to develop, and has a posterior end, which is where the tail's going to develop. Now, we're humans, we don't have tails, but at the embryological stages, there's not a whole lot of difference between what's going on with the human embryo and other mammalian embryos. So the human embryo does have a tail, so to speak. We just doesn't persist into the, even into the fetal stages. By then it's gone. But this end down here is the tail end. The legs grow out of this end of the embryo, but they don't really extend out. They kind of, the fetal position is really describing how the upper and lower limbs kind of wrap around underneath the fetus as it's developing. To look at how the nervous system develops, this figure down here shows two time points in fetal development. The first one, which is about 3 or 4 weeks into development of the embryo, the anterior end of the neural tube starts to swell out in a few places, giving rise to what we call vesicles. And there are 3 of them in this primary stage, which are named as they're labeled here. These are Greek words: prosencephalon, mesencephalon, and rhombencephalon. Encephalon, which is common to all these words, in Greek means in the head. And so that basically means the brain. And then pros means forward, mes means middle. Rhomb does not mean hind, but keeping in the same sort of construction here, we have the forebrain, midbrain, and hindbrain regions. The forebrain and the hindbrain then develop a little bit more deep definition as they separate into a couple of vesicles each. The midbrain doesn't really change at all. And in fact, when we get to the adult brain, there's still a region called the midbrain, which is essentially this part that is practically a third of the early embryonic brain, but it's a very, very small part of the adult brain. It just happens to be this same part as it develops along. the forebrain becomes these two regions here called the telencephalon and the diencephalon. The telencephalon gives rise to what we call the cerebrum, which is, when you think of the brain, it's probably the thing that you usually think of, the big, wrinkly outer part. So in this hard plastic model, this kind of outer pink part here, this is the cerebrum. Now, as I'm holding this, I'm specifically covering the sort of tan, wrinkly part here because it's not part of the cerebrum. What we usually think of as the brain, or we see the most of, is the cerebrum. And cerebrum is really just the Latin word that means the same thing as encephalon. And then the diencephalon becomes a number of different structures, which we don't have really a simple, inclusive name for that. So we still call that the diencephalon. When I was learning this in graduate school, one of my teachers just described the diencephalon as anything that has the word thalamus in it. So the major part of it is what's called the thalamus. But then there's a couple of other structures that are listed here that have thalamus in them, the hypothalamus and the epithalamus. And there's a couple of other thalamus things which we won't really deal with too much. But basically, if you're reading anything and it has thalamus in its name, you can assume that it's part of the diencephalon. Diencephalon also has, at the embryonic stage, the eye cup. And this is what's going to become the nervous tissue in the eye. It's going to become the retina. It actually develops out of the central nervous system. We usually think of the eye as being a peripheral structure because it's outside of the brain. It's outside the cranial space. But it's actually, embryologically speaking, part of the central nervous system. It just had some bone grow around it, separating it from the rest of the central nervous system. But for most purposes, we usually just think of it as peripheral because it is outside of the brain. But the fact that it's in the diencephalon in the embryonic brain is useful because what we'll find is that the eye actually connects directly to the thalamus and the hypothalamus, which will be helpful to understand how some of that works. The hindbrain becomes two structures, which are, their Greek names are here, the metencephalon and myelencephalon. The metencephalon becomes two structures, which are basically the same point, the pons and the cerebellum. And then the myelencephalon becomes what is most formally known as the medulla oblongata, although we'll only ever call it the medulla because there's nothing else with medulla in its name that we have to differentiate this from. So if I'm talking about the medulla, this is what I'm talking about here. So that's just a quick sort of run through of what the figure here is showing. And it shows us the different structures that are going to make up the adult brain. When we talk about the adult brain, we usually think of four different structures. There's the cerebrum, which I already described for you as most of what you think of as the brain, the big group, the outer part. That's not the only thing that's the cerebrum. In the picture that we're looking at here, pictures that we're looking at here, the wall of these embryonic brains are just a thin layer of cells. But those cells are going to develop and become thicker and thicker, and axons are going to grow through the area. And so there's going to be white matter and gray matter and all this stuff in there. And it's going to become a rather thick structure. So if we were to look inside this model, which we can't, we'd see more inside of the cerebrum. Okay. And then the diencephalon, like I said, is anything with thalamus in its name. That's a separate section of the adult brain. And then we have the brainstem, which is the midbrain, pons, and medulla all together. And then the cerebellum on its own, which is the other wrinkly part. So as I open up the model here so we can look at half of it, just half of it, we can see all these structures represented. So again, the cerebrum is this big outer structure you see here that has a bunch of wrinkles all over it. Towards the center of the brain, there's this, well, at least in the hard plastic thing, a white sort of C-shaped structure that's at the edge of the wrinkly part. That's the edge of the cerebrum. What's sort of inside that C-shaped structure, which in the hard plastic brain, there's a little blue thing, a red thing, and a yellow dot, and then some more pink colored stuff. That's essentially where the diencephalon is. Now, if you have the hard plastic brain, there's a yellow dot in the middle of that. If you have the soft plastic brain, or the soft rubber brain, whatever, it's a brown dot. Okay. That dot is right in the middle of the thalamus. The two sides of the thalamus, what are the two walls that we see here, as they get thicker and thicker, they grow in towards each other, and they actually touch a little bit in the middle. There's a little gap between the two sides of the brain, and we'll talk about that space in a little while. But where that yellow or brown dot is, it's where the two thalami, which is plural of thalamus, actually grow so close together that they touch just a little bit in the middle. There's not really any significant connection from one side to the other right there. They just physically touch in the middle. Unfortunately, somebody has defaced the model that I have here by taking a ballpoint pen and drawing in the groove that's just below the yellow dot on this. Hopefully, yours hasn't been defaced the same way, but that ridge there is the ridge that separates the thalamus from the hypothalamus. So hopefully you can see that in whichever model. So the little brown dot that you have right in the middle there, there's that brown dot's in the middle of a kind of oval structure. Mm-hmm. So the ridge that separates that from what's anterior and inferior to it. Okay. That ridge separates the hypothalamus from the thalamus. And the hypothalamus is the pointy part that kind of comes to a corner right at the edge of this cerebrum that we can see here. You might have in your model a little peanut-sized thing attached by a thin, call it a wire, underneath the hypothalamus. If you have that little peanut-shaped thing is the pituitary gland, which is attached to the hypothalamus. There's only one pituitary gland, and so it's only going to be attached to one of the two halves of your brain model, and not every brain model is going to have it. So chances are you're not looking at a half of the brain that has a pituitary attached to it. But if you are, that's another clue as to where the hypothalamus is. That's the diencephalon, a little bit right in there. And then what's extending away from the diencephalon down, like a stem of the brain, is the brainstem. There's the medulla, the pons, the midbrain, the pons, and the medulla going down from the diencephalon. The big oval structure that's kind of halfway down the brainstem, that's where the pons is. Now the pons is that oval structure and what's adjacent to it. And actually, I think I have that. Okay, so, well, here we go. Here's a mid-sagittal view of the brain here in the book. What I've got the mouse on here, that's the dot that's either yellow or brown in the model you have. And there should be a ridge right about here separating the thalamus from the hypothalamus that comes to a little point right here. And then the midbrain is the top part of the brainstem that ends where that oval is. And that oval represents where the pons is, which extends back through the whole width of the brainstem. And then below below the oval portion is the medulla. Okay. So that just lays out the three parts there. And the easiest way to kind of see where the three parts are is the oval structure that we see in cross-section here is the pons. Okay. Now, what's not in this picture, what would be attached to the posterior side of the brainstem, is the cerebellum. Okay? And the cerebellum is highlighted down here. This is a picture that highlights the structure of the cerebellum in a little bit more detail that we used in the book instead of just another one of this sort of thing. The pons is where the cerebellum is mainly attached, and the pons and the cerebellum grow out of the same part of the embryonic brain. So it kind of shows us a little bit there. Now, one thing that's useful about thinking about the structures of the embryonic brain is to understand how things are connected to each other. So like I said, the cerebellum and the pons both grow out of the same area here, and the way that the cerebellum is attached to the brainstem is mainly through the pons. There's a little bit of an attachment to the midbrain and a little bit of an attachment to the medulla, but that's it. There's no direct attachment from the cerebellum to, say, the cerebrum, because obviously they're quite far apart from each other. When you look at the adult brain like this in the model, it seems as if the cerebrum and the cerebellum are right next to each other, but they're not. So just because you see that they grow together within the space of the skull doesn't mean that they're connected to each other. How we see things laid out here tells you how things are connected to each other. If part of the cerebrum wants to contact part of the cerebellum, it has to go through all the intervening parts. Even in the model here, where the cerebellum and the cerebrum are right next to each other, a millimeter apart from each other maybe, any information leaving the cerebrum going to the cerebellum has to go through the diencephalon, the midbrain, and into the pons and into the cerebellum. And anything from the cerebellum has to go back through the brainstem and the diencephalon into the cerebrum. So even though they're physically right next to each other in the model, there's no physical attachment point between them, separated from each other. And the thinking about the embryonic brain really reinforces that understanding. I mentioned a little bit about the eye cup too. That's the retina. And when we think about the adult connections of the retina back into the brain, primarily it's attached to the thalamus and the hypothalamus. What we usually think of as vision or conscious perception is handled by information from the retina going to the thalamus. And then the thalamus sends that on to the cerebrum. Our unconscious visual processing is handled mostly by the hypothalamus. And being unconscious, you're not aware of it, but if bright light hits your retina, your pupils close down to protect your retina, and that's handled by the hypothalamus. When it's daytime, sunlight hitting your retina tells your body that it's day, not night, and the hypothalamus is sending that information on to other parts of the brain. So the hypothalamus is receiving that. There is actually a little bit of an input from the retina directly to the midbrain, but they're right next to each other, so it's not very hard to see that happen. And actually in the structures of the adult brain, they're actually, the part of the thalamus that receives retinal input is right next door to the part of the midbrain that receives it. So it's not a big stretch for it to make that connection. But it can't go that far away. It has to go somewhere nearby. There's no direct visual input to the cerebrum. So when we think of our conscious perception of vision, that's taking place in the cerebrum, but it's getting that second hand through the thalamus because the eye cup's attached primarily there. And it comes from understanding this embryonic connection here. Now, the original tube was, actually, I think I closed that picture up. But let me just go back to the picture we were just looking at here. The tube that we see in these series of embryological sections is hollow down the middle. And then even with the vesicle stage, we can see the wall that's the outside of whatever encephalon region that we're talking about. But the center is actually hollow. Another thing to get out of the embryonic brain is to understand where these spaces come from. Now, these spaces that we see in the embryonic brain, in the adult brain make up a system of empty spaces that we call the ventricles. And here I have a section of an actual human brain. And you can see in the middle, kind of like a Y structure, there's some empty space inside the brain. That's what's left over of the empty space of the embryonic brain. And that Y structure that I'm pointing at there is basically a Y coming off of either side of the cerebrum and then coming down the middle through the diencephalon. Those ventricles have formal names. There's 4 ventricles. The first 2 are referred to as the lateral ventricles. And the picture for this is in the circulation and the central nervous system part of the book. Oh, I don't have a picture of that. Okay, I thought I had a picture to show you, but no. This is what the other model I asked you to get, which Tiny's the only one who got it. This model right here. Okay. This is what's called a ventricular cast model. Oh, sorry, Chelsea, I see you have it here too. Oh, you guys got it. Okay, you guys got it. Great. There's 3 up here, so I'm trying to figure out who didn't get that. But this is a ventricular cast model. These are made by taking a brain like this and injecting plastic into these spaces in the middle. Okay? And so the plastic solidifies, filling up all of that space. And then the tissue is removed from that, and what's left over is a shape like this. Now, these actual models that we're looking at here aren't primarily ventricular casts. These were made from a ventricular cast that was developed the way I just talked about. The 2 big kind of pointed C-shaped things here on either side, those are the lateral ventricles. It's ventricle 1 and 2, the first and second ventricles, but we don't number them anymore. We just refer to them as a pair, the lateral ventricles. And then in the center is the third ventricle. There's a little hole in the middle of this, which is where that yellow or brown dot would pass through. The third ventricle is in the diencephalon. So where the hole is right there, that's the third ventricle that's in the thalamus region. And then where it comes down to a point here, that's where it's down the middle of the hypothalamus. You can kind of imagine that you're looking at the head with an eye and a beak kind of coming to a point here. So if you can kind of imagine that's the head of something. Coming off of that head, what would be the neck is what's called the cerebral aqueduct. It's not a ventricle itself. It's just what represents the connection from the third ventricle in the diencephalon to the fourth ventricle that's going to be down in the pons medulla area. And so then where that thing opens up, the neck structure kind of opens up into, I don't know, kind of a scoop looking thing. That's the fourth ventricle. Now you can see most of this if you look at the model. I say most of this because you can't see the lateral ventricles. They're inside the cerebrum. But if you can imagine the shape of the ventricular cast superimposed over the brain here or like this, you can kind of imagine where it would sort of sit in the middle. And there's a I guess it was in that previous section. Yeah. So the dotted line that we see here That's the corpus callosum. Sorry, the white C-shaped structure there. And the lateral ventricles are located sort of right above that, sort of off to the side. And they follow along in the shape of the brain. It comes to kind of a point in the back, and then it goes into this side over here. So you can't see the lateral ventricles in these models because you can't see inside the cerebrum. But the third ventricle would be all of the space around here. You can see the yellow or brown dot, depending on which model you have. That would be the hole through the middle of the third ventricle. And where it comes to a point is down here where the hypothalamus comes to. And then you can see a little ridge going through the midbrain region, and that's the cerebral aqueduct. And it opens up into some space between the cerebrum, between the cerebellum and the brainstem. And that's where the fourth ventricle is. Those are the ventricles you can see. In the section that's circulation in the central nervous system, it talks about the ventricles. It also talks about cerebrospinal fluid that flows through that space there. And I'll let you get that out of the book. I want to move on to talking about the structures of the cerebrum in more detail. So this is the next section after the embryonic perspective, just about the structure of the central nervous system. So just before I talk about the cerebrum in more detail, it starts off with the cerebrum and then the diencephalon and the brainstem and the cerebellum. So there's 4 different regions of the adult brain are just laid out as we go through this. What I want to do today is just concentrate on what we see in the cerebrum. And what we can see in the cerebrum are the patterns of wrinkles and folds, which we can use as landmarks to find our way around the brain. Now, the first and easiest thing to see is the longitudinal fissure, which is the separation that's right down midline between the right and left sides of the brain. It separates the cerebrum into what we call the right hemisphere and the left hemisphere. And if we were looking at actual human brains here instead of these models, we could put a probe or our fingers or whatever down into the longitudinal fissure and actually pull the 2 hemispheres apart a little bit. The first place we'd actually run into any kind of resistance would be this big C-shaped structure right in the middle, which is called the corpus callosum. And for the sake of you saying that term, let me go here. So where this dotted line is, that's where there's this structure called the corpus callosum. In the hard plastic brain, it's this white C-shaped structure here. In the soft plastic brain, it's more of a light gray color. That represents axons that are coming from one side of the brain and connecting to the other side of the brain. So how the right hemisphere and the left hemisphere communicate back and forth with each other is by the fibers of the corpus callosum. Now, I want to show you a video in a second that demonstrates sort of how the corpus callosum helps the brain function, how the 2 sides end up communicating with each other. Well, let me just play the video because it explains what's going on. If you want to see this video again after I show it here, it's in the video section on Blackboard under video content. I put a video in for the, I embedded the video that we watched before about the electrical activity across the cell membrane, which is one of the videos I showed you. And then there are a number of other ones in here that I've put that might be of interest. I like to use this video here, and I want to play it for you. Now, for any of these videos, they're just YouTube videos that I've embedded here. Within Blackboard, I have a way of embedding YouTube videos. You can watch them directly on the page here, but if you want to go to the YouTube page and you can enlarge the screen or whatever, the title of the video up here at the top, that's an active link to take you to YouTube, or I guess you could just click on YouTube there. Here, let me turn more lights on so you can see this better. This is part of the PBS Special. Turn this up. So this is Alan Alda. He's at Dartmouth in New Hampshire.
Mike Bizonica, and a man he's worked with for over a decade. A man with 2 brains. You've been working a long time. Dr. Bizonica, 14 years. It doesn't seem like that now, does it? Hold on now. The collaboration began when Joe had surgery, and you had this procedure to correct that epileptic problem. Is that right? Tried to stop the seizures. I was having seizures like every day, so there's 2 or 3 a day. To control Joe's epileptic seizures, a surgeon severed the connection between the 2 halves of his brain. Cutting the corpus callosum like this prevented the spread of the electric storms that caused the seizures. But it also prevented the left and right halves of his brain from communicating with each other. In the years since the operation, Joe's epilepsy has been under control. He now earns a living at an egg farm, and in his everyday life, he's largely unaffected by the fact that his left and his right brains work independently. Do you feel any different when you think about something than you did any differently from what you felt before the procedure? Better back up, right? That's something everybody could use. I found out how true that was right away when I was asked to draw a different shape with each hand. In a brain like mine, roughly speaking normal, at least all in one piece, the left half of my brain controls the right side of my body, while the right brain controls the left side. Oh, no. But because the 2 halves are connected, nothing wrong with that. Getting each hand to work independently isn't easy.
Well, we're saying that the fact that Alan's hemispheres are connected, and the motor messages from one are confusing the motor messages in the other.
I was just drawing an upside down duck. Okay. But when Joe was given the same task, his 2 hands operate as if controlled by 2 separate brains. What's happening is that each half of Joe's brain is given a separate instruction. He's asked to fix his eyes on the cross in the center of the screen. Anything flashing to the right of the cross goes only to his left hemisphere. Things to the left go to his right hemisphere. Because the 2 don't communicate, each hand does only what its half of the brain sees. Wow. It's really like 2 different people doing the same.
That's right.
Okay.
That's the idea. Okay, Joe, I want you to keep your hands up.
In an experiment that's now a classic in brain research, Mike Bizonica over 30 years ago used a similar setup to find out if the 2 halves of the brain are specialized to do different things.
Sure.
Joe is being flashed a word only to 1 half of his brain. Words flashed to the right are seen only by his left brain. And Joe can report seeing those words just fine.
Good.
But when a word is flashed to his right brain,
You can see that.
okay?
So I'm going to ask you
But now watch what happens.
to draw that with your left hand. I think I'm lost. Why don't you try drawing another picture of it over here if that'll help you. Oh, okay.
It's almost as though somebody has given him a secret communication. That's right. Now he knows that it's up until then he was blind.
Exactly.
When Bizonica first did this experiment, it instantly proved that the ability to speak resides almost exclusively in the left hemisphere. Not until he sees what his right brain is drawing is Joe able to name it. He said church after looking at it. But he had to figure it out about as long as we did. That's really interesting. It's a picture here of somebody communicating almost with another person.
Communication is not occurring inside the head, it's occurring out on a piece of paper.
Yeah.
Wow. I don't know. You want to draw a little bit more?
So far, Joe has been seeing only one word. Things get even stranger when he's flashed two words, each to only one half of his brain.
The right hemisphere is on toad.
Yeah. And so his left hand draws a toad.
So there's the toad.
Oh, it's a toad. And this time I was able to guess what was coming.
Now put a little three-legged stool in there later or what?
Joe's speaking left brain saw a stool. Saying the word lets the hand that's controlled by his right brain in on the secret. That's great. That's really interesting. And if he had seen that with the corpus callosum intact, he would have drawn a toadstool.
Right.
Yeah, not the toad and the stool.
Right, exactly the point. I've been doing this for 35 years. Yeah.
And it
Gets me every time.
Yeah.
Okay. So there's actually a second part to that, which you're welcome to watch if you want to. Now, it explained in the video there kind of what's going on. The patient, Joe, there has had this procedure done because he had really bad epilepsy. And activity would have started in one spot in his brain somewhere and spread out through the rest of his brain and caused a seizure. And like he said, it was happening pretty much every day. And that can be really debilitating. He couldn't probably have held down a job room because he probably would have started having grand mal seizures and lost control of muscle control and that sort of thing. But by severing the corpus callosum and keeping the information, or not the information, the activity that's originating with epilepsy from crossing over the other side, it limited the spread of the or the extent of the problems he was having with it, and he was able to start having a normal life. One thing I really like about this video is it has that little sequence where you can see him driving a truck, working at an egg farm, that sort of thing. He leads a normal life. You know, little interaction, he's talking. If you'd met that guy yourself on the campus at Dartmouth, you wouldn't know that he had a corpus callosum split. It's just, you have to get into the activity a little bit deeper, into the function a little bit deeper to really see what's going on. And so they did a couple of
also prevent
different tests. So here, and Alan Alda being the normal so-called control here, is being exposed to two different things, and he has to draw those pictures in front of him. Okay. He can't see the paper where he's drawing, so he's relying solely on his control of the two hands to do what he's supposed to do. So he sees this flashed on the screen in front of him. He's fixed on the little cross in the middle. That's the fixation point. His eye's not moving from that. And the images are flashed up there so quickly that he doesn't have time to move his eyes. And they had a little demonstration in there that one image is projected entirely to the other side of the brain and vice versa. And then he's controlling his two hands because the two sides of the brain control the two sides of the body separately also. The left side of the brain controls the right hand. So drawing with the right hand is coming exclusively from input from the left side of the brain. It's more than that. It's not just being able to draw with that hand. It's all movements on the right side of the body, moving arms or legs or anything. It's controlled by the left side of the body. All sensory input, anything that you touch with your fingertips, or something brushes against your thigh or whatever, that's all sent to the left side of the brain for you to process. Vision's a little bit different. It's not what the right eye sees is sent to the left side of the brain, but rather the right side of visual field. So everything from that point in the middle over is processed entirely on the other side of the brain. And so what his left brain sees, the right visual field, he's able to draw with his right hand. And then what his right brain sees, left visual field, he tries to draw with his left hand. Now, Alan Alda, as the sort of control here, isn't able to do that because he has an intact corpus callosum. And that's what's happening for most people, is that our brain's communicating back and forth. And so what's processed on one side of the brain gets shared with the other side of the brain. And he can't separate out those two images, and the command to make his left hand move interferes with the command to make his right hand move, and vice versa. And so he's not able to quite draw the pictures exactly how he sees them. Joe, on the other hand, doesn't have that interference. He's not getting commands from one side of the brain going to the other and interfering. So the two hands can work independently and generate those two different images. Then they moved on to the word presentation thing. And in the video, they start talking about how language, or I shouldn't say language, speaking is handled almost exclusively by the left side of the brain. Now, probably 90 to 95% of the population, the left side of their brain is responsible for producing speech. That doesn't mean that the left side of the brain is the language side of the brain. And in fact, this video disproves that. Because his left and right brains are not connected to each other. Or I should say left and right hemispheres aren't connected to each other. But he's able to understand language and use that to draw something with his left hand. So for instance, to put it the other way, like the word phone was flashed up on the left visual field. So his right brain saw that, but he's not able to verbally say the word phone because the part of his brain that makes speech isn't in the loop. But he's able to draw a picture of a phone with his left hand, which represents what his right brain saw. And once he saw that and recognized it as a phone, which it took a few tries to even draw something that resembled a phone, that the left side of his brain could say, oh, that's a phone, that he was able to say it. And then when the words were flashed up, both on either side, toad and stool, he was drawing exclusively with his left hand. So it was just the right side of his brain doing it. So the right side of his brain saw the word toad and drew a picture of a toad. And then he said, I saw the word stool, because the left side of his brain processed that on the right visual field. And saying the word stool let the right side of his brain know that he also had to draw a picture of a stool. So the right side of his brain heard the word stool and was able to interpret it. It just couldn't produce speech to say the word stool. It was able to draw the picture of the stool. So there are language functions that take place on the right side of the brain. It's not exclusively language on the left side, but the speech production stuff is mostly there. And when he's talking about at the beginning, it's like having an extra brain. What he probably really means there is that the left side of his brain, left hemisphere, is really his only conscious perception of the world. When he's talking about what the left side of his brain is experiencing. He doesn't talk about what the right side of the brain is doing. And so I'm sure there are certain instances where he does something with the right side of his brain that he wasn't expecting, because the left side of his brain, which is where his sort of conscious perception and internal voice resides, wasn't expecting it there. But I wanted to show this to kind of set up, to begin with, the idea that different parts of the brain do handle different functions, whether it's left versus right control of the body or producing speech, those sorts of things. There are different parts. Now, I do need to qualify something here, or I need to deal with something that people have probably heard of. Even in the links here, there's another section of the video here with Alan Alda and Dr. Xanaga that's titled Right Brain versus Left Brain. And it's from this work that Dr. Xanaga has done up at Dartmouth that a lot of people have gotten this misconception that we have a right and a left side of the brain that's in charge of different things. There's, I think it's a couple of years past now, but there's a set of Mercedes ads that sort of use this. You might have seen these. Come on, where are they? There we go. So they have this picture of the brain. They got the left side, and it looks all kind of clean, and it's got, let's see, this version one, you can't quite read what's written in there. But there are other ones that have like math equations and stuff like that written in there. This, I think, is computer code. It's typed over here. And then the right side has all these colors, and it's bleeding out past the lines, that kind of thing. This idea that we have a left brain and a right brain is actually a very false impression that comes from the work that Xanaga has done here. People can explain things. People that have had this split brain procedure done, cutting corpus callosum, can explain things based on what the left side of their brain is doing. And the right side of their brain seems to be nonverbal. And so somebody took that a little too far and started to say, well, the left side's the analytical side and the right side's the artistic side and that kind of stuff. It's not at all true. Okay? It's really a misunderstanding of the work that video I was just showing you comes from. Okay? So, all right, back to the picture of the book. So the first thing we can see in the brain is really the two sides there. Okay? The longitudinal fissure separates us into the right and left hemispheres. Another video, which I'm not going to show you, you might have noticed the thumbnail after the one I just showed you finished. This girl here, the title of this is Girl Living with Half Her Brain. I don't entirely remember the story clearly, but if I remember correctly, this girl, when she was probably a newborn or 1 or 2 years of age, had a rather severe encephalopathy or encephalitis or something like that. And so to remove the problem, they actually cut out half of her brain. Now, doing it so early in her life, it didn't cause problems because her brain was still growing, so it just grew into the space that was freed up by removing the rest of the brain. And she probably functions very well. You can watch this video, and she's being interviewed here on the Today Show. And she's probably an adorable little kid, and you wouldn't know necessarily that she has just one hemisphere of her brain. It's just that it's grown into all the space there, and it's taken over a lot of the functions that the other half of the brain would have been responsible for. Because just half the hemisphere has been removed, so the other half just grew in and took over. For other people, the brain is very predictable. The way that it grows into the space of the cranial cavity is pretty consistent. We can look at the surface of the brain and pick out some landmarks that'll tell us something about where we are. And so this picture here starts to get at the broad ideas of where we can see things. So on the model that you have in front of you, the easiest thing to see after the longitudinal fissure is this groove right along the side. This is called the lateral sulcus. Now the word sulcus means groove. Okay. So here's the label, lateral sulcus. It's this groove right here. Any of the grooves that we see in here, where two folds come together, those are all sulci, which is the plural of sulcus. It just this lateral sulcus is the most obvious one. It outlines what we call the temporal lobe and separates it mostly from the frontal lobe up here. But the lateral sulcus actually goes back a bit and separates the temporal lobe from this other part here called the parietal lobe. There's another sulcus that separates the parietal from frontal lobes. If you're looking at the surface of the brain, you should see three sulci, three grooves that kind of go in parallel down the side. One of them should go all the way from the lateral sulcus to the lateral fissure, said it wrong, longitudinal fissure, almost to the lateral sulcus. And then there should be one running sort of parallel to it and just anterior and one just posterior. The two wrinkles that those three grooves define are called gyri. Gyri is plural for gyrus. And so all of those wrinkles that we see there are where the brain's folded up to fit into the space there. The center of those three sulci running down the length there separates the frontal from the parietal lobes. It's called the central sulcus. From the central sulcus to the next anterior sulcus right here, that gyrus is called the precentral gyrus. And then from the central sulcus to the next sulcus over, right about there, which is not always that continuous, that next sulcus there, a gyrus there, is called the post-central gyrus. Okay. So the precentral gyrus is the very back of the frontal lobe, and the post-central gyrus is the very front of the parietal. As you're looking at your model, it'll be labeled. You'll be able to find where that central sulcus is. You have the hard plastic brain on one side, and both of these types of models are only labeled on one side. We see the number 4, okay? Sort of right in the top center of the brain. The sulcus that's just before the number 4, that's the central sulcus there. So once you've found that, you can kind of imagine there are two gyri running alongside that, okay? Which are the pre and post central gyri. If you have the softer, squishy brain, there's a number 5 with an arrow on it pointing to the central sulcus. And then the precentral gyrus and the postcentral gyrus are both labeled, I think, numbers 6 and 7. So you can see that really clearly there. But from those two landmarks, we can actually separate the cerebral surface into 3 major regions: the temporal lobe, the frontal lobe, and the parietal lobe. Those lobes are obviously all named after what? What's also called frontal, parietal, and temporal? No, it's the bones of the skull. Okay, there's a frontal bone, there's a parietal bone, there's a temporal bone. There's also an occipital bone, and there's an occipital lobe. The lobes of the brain are named for the bones that cover them, or the bones are named for the part of the brain that they cover. The names go together. Now, there's not a landmark that separates the occipital from the parietal and temporal lobes that we see here. The only way that we can actually see that is if we go to the medial surface of the brain. So if you look at your model on the inside, there should be a nice significant groove or sulcus going up sort of from where the cerebrum is straight up. Okay? And that's what's called the parieto-occipital sulcus. That's labeled here on the screen because it goes over the longitudinal fissure just a little bit. So we can see it on the lateral surface right there up where the label is. but it ends. From that point on, there isn't any specific landmark that we see there. Okay? Now, as we're thinking about function of the brain, so before we were talking about the left side of the brain for most people is responsible for producing speech, that sort of thing. We can get into more and more specific functions on the surface associated with different parts of the brain. And so the lobes we tend to think of as having particular functions associated with them. The frontal lobe is most often thought of as the motor lobe. The precentral gyrus, the cells that are found there, are responsible for causing muscle movement. So when you decide to move part of your body, the command coming from your brain to make that voluntary movement is coming from the precentral gyrus. Anterior to the precentral gyrus is a region that's sometimes called the premotor area, and it's responsible for planning movements. So when you decide to move something, something a little bit more anterior in the frontal lobe is responsible for telling the precentral gyrus to tell the muscles to move. And as we move forward in the frontal lobe, we get into more what's what I'm looking for here? Well, it's hard to explain. I'm losing the word I'm trying to say here, but we move from actually making movements to planning movements to thinking about movements to deciding to move to interpreting whether we should do something or not. And when we get up to the very front of the frontal lobe, what we call the prefrontal area, we often associate that with personality. And that's kind of in the sense of you are what you do. So if you decide to take a nap in the afternoon on Mondays and you don't come to class, okay, that's going to set up one series of events, which might be that you don't get to come into class here and you don't pass the class. Okay, that's not deciding to get out of bed physically to stand up, but it's sort of the precursor to that. It's like, I'm going to sleep. And then you never get around to deciding to get up. But it leads into the motor responses that would, well, not getting out of bed is kind of a negative motor response. Right? That's the idea. What you do largely defines sort of who you are as a person, your personality. And that's a really kind of vague way to put it, but there's good evidence of different regions in the prefrontal cortex that's responsible for thought processes. There's a study I read about a year or two ago that it's a very simple kind of psychological study, just asking people if they want to have a brownie or a cookie. Okay? And they had to choose whether they wanted a brownie or cookie. And then I think subjects in the study were given a chance to talk to each other. And what they found was people that had a very strong opinion over whether they wanted a brownie or a cookie had a fairly active region in the prefrontal cortex, which apparently leads to having opinions. And people that were kind of iffy about things, that part of the brain wasn't as active. So again, that's kind of getting at personality type issues there. Now, motor is sort of one half of the major functions of the brain, motor versus sensory. So if the frontal lobe is responsible for motor function, basically the rest of it's about sensory function. It's just that the three lobes we have left, parietal, temporal, and occipital lobes, we associate with different aspects of sensory function. The occipital lobe is largely thought of as being the visual center of the brain, which is true. The beginning of conscious perception of vision starts with the occipital lobe. And then the parietal lobe, we often associate with what's called somatosensation, which is our sense of touch and other aspects of how our body interacts with the world through senses. When we say touch, we mean a lot of things. We mean pressure and vibration and temperature and pain and those sorts of things. So all of that, as well as our feeling of our body when we move our body around in space, or the position of our body, all of that's handled in the parietal lobe. The temporal lobe, some sources will associate it mostly with hearing, which is actually a very small part of what the temporal lobe does. More importantly, the temporal lobe is responsible for memory. And memory is really a sensory function. When you remember things, you're remembering how things felt or smelled or sounded or tasted or those sorts of things. You're remembering the sensory experience. So memory is really a sensory experience. Now, I have sort of a story to go with memory function there. I want to save that. We'll take a break, and then we'll come back, and I'll come back to the memory thing. And then I'm doing pretty good time-wise today compared to what I do with my night class. So I'll probably be able to get to the cranial nerve stuff, which is what the other lab quiz is in this week's folder. And I'll present that information for you in the second half. So let's go ahead and take a break. Be back in here about a quarter after or so. Take about 20 minutes. And we'll get started and we'll finish up this neuroanatomy stuff. And I'll touch on the cranial nerves, which is the lab material for today.